Welcome, everyone, and thank you for joining this call. It's been such an exciting time in our Duchenne community with the approval of Sarepta's exon dys 51, or eteplirsen, better known as eteplirsen, and the news about the partnership with Catabasis, and most recently, the partnership with Sarepta. So today we're here to discuss with Summit. We're here to discuss that partnership with Ed Kay and with Glenn Edwards from Summit to discuss the partnership and learn more about it. So with that, Ed and Glenn, you're on the phone, I'm going to assume. Is that right?
Yes, that's right.
Glenn, but wasn't a bit unsure of that. Okay, so we're going to start with some big overview questions, if you don't mind. And so the first question is, why did you do this deal, and how was it structured as a partnership?
Well, I can start if you want, Glenn, and then I'll certainly let Glenn comment on this. I think for us, it gave us an opportunity to work with a company that has a great deal of experience with utrophin upregulation, and so it was an area of expertise that we didn't have. We had already been doing some collaboration with Summit before about harmonizing biopsies and things like that. So I think for us it made perfect sense to have a partner who has a lot of experience in muscle biology, especially in utrophin upregulation that we did not. And I think for looking at the potential of combination therapy, and really Summit was at the forefront for all of the companies working in utrophin, in the utrophin area. So I think it made sense if there was some ability for us to work together. From Sarepta's point of view, it made perfect sense. Glenn?
Yeah, I agree, Ed. I think there are two factors. One is the sort of cerebral one, which you've pointed out, that you've got a huge amount of experience in development of treatments for DMD with your recent regulation, but also the clinical trials that you've carried out. So for us, getting access to that expertise was really important. But on a more emotional level, the parent groups, PPMD included, have really encouraged the companies to work together on non-competitive things like biopsies and what are the correct endpoints. And so we've worked with your team, over the last 18 months on a number of things. And there seemed to be such a great fit between the companies, the collaborative nature of doing things. So we felt not only at the level of the facts and the figures did it make sense, but also we felt the teams could really work well together to deliver these really important treatments to the patients.
Thanks, Glenn, and thanks, Ed. And I think, we were very excited about this combination, clearly because the Summit drug target is the promoter for utrophin, so basically increasing utrophin and getting it across that membrane and re-expressing shortened dystrophin should be theoretically really amazing for all of the boys whose mutations are amenable to the exon skipping technology, and all boys are amenable to the utrophin target. So what does this mean for families in the U.S. and Europe? And I ask this just from the standpoint of often, or in our limited experience when we've seen companies join together, sometimes we see a delay or we see some changes in the clinical trial plans. So I wonder if you each could speak to that for us, both in the U.S. and Europe. And maybe start with Europe first, since you have, Glenn, been in studies in Europe, and then we'll take on the U.S.
Yeah, I think it's really quite important that from the get-go, it was clear that we didn't want to see this collaboration resulting in delays. What we wanted to see over time was getting more good decision-making and a wider group of people involved in the future development, but that we would carry on with our current development plans. And as you're aware, Summit has got a phase 2 study recruiting in the UK right now with US sites about to come on imminently. So I think in the short term we won't see certainly any delays as a result of this collaboration. And in the long term, I would hope to see a better decision-making and therefore a more effective development path coming out of it. And I think it's important to say that, the expectation is that for the development phase, it'll be Summit that's carrying out the work. So there's no kind of handover period envisaged. What we'll see is a seamless development program with initially Sarepta providing expertise through a joint steering committee, but the Summit team will actually execute these trials. So I think the short answer is there should be no delays, and I would expect to see in the longer term a more effective development plan coming out of this.
Thanks,
I would second that also, and I think one of the things that we looked at Summit as a partner is that they were prepared to do the development themselves, and it wouldn't distract us from our commercialization and also our other development plans for next generation chemistry and follow-on exons. So I think that's what made it very important, and if anything, we hope that it strengthens the possibility for long-term development, but certainly nothing that would obstruct or delay any of the trials.
Thanks, Ed. That's very helpful to know. And actually, it sounds like a terrific model where you're both not independently but in parallel moving forward with your compounds with the goal of expediting those as well as thinking about combinations. So just to build on what you were talking about, Glenn, one of the things that I think has been expressed by concern of the European families, I know that the Summit drug, Uzotromid, is that how you say it, was now reformulated and is more bioavailable, and that compound is referred to as F6. So in the U.S. and in Europe, will you be using this new reformulation?
No, that's a really good question. So we do have two formulations. There's the current formulation known as F3, and that's what's in the phase 2 study at the moment. And we think that is likely to be effective. It is likely to upregulate eutrophin and will give benefit to patients. That's our expectation, and that's why we started the phase 2 study with it. But we're not content with the state of development of the drugs now, and so we've had several things going on in parallel, including an effort to see can we get even better formulations of Uzotromid, and as a result of that, we have just recently announced some really exciting data with a formulation that we call F6. And in addition, we're working with the group at Oxford University on completely new molecules. So, longer term, we would expect to see even better eutrophin modulators coming through. But let's just focus on your question, which is F6 and F3. Certainly, the initial data on F6 indicates that it is more bioavailable, and from our preclinical modeling, that should upregulate eutrophin even more. And so, 10 boys, likely at this current state to be in the US only, will get the F6. And what that will do is, over the course of the study, when we get the data, allow us to really evaluate both the efficacy data of the two different formulations and the safety data of the two different formulations, and really establish from a factual data driven basis which of the two is the best for long-term treatment. And when the study finishes, we hope to then be in a position that we would have enough material, both of the F3 and the F6, to be able to then go into future studies with whichever of the two turns out to have given the best combination of efficacy and safety. So that we think both of them are likely to give some benefit, and we will make the decision on which to progress with based on what we actually see in this phase-out EMD study. Our intention, obviously subject to regulatory approval, is not just to start new studies with whichever is the best formulation, but our intention is also to get material so that the boys in the current study could continue to receive drug, whichever of the two turns out to be the best. So we plan, if you like, an extension study to the phase out DMD, so that at the end of dosing, boys can continue to receive whichever of the two has given us the best data. That's obviously, subject to regulatory approval. And I can't predict which of the two it will be. But if you've been on F3 and it turns out that F6 is the best, then you would move on to F6. And if it turns out that the other way around, then we would have supplies of both, so that the best one can be what boys continue to receive, and also the best one will be what goes into future clinical studies.
Right. So just to clarify then, once you do that 10 child study or 10 young boy study in the U.S. on the reformulated F6 compound, you'll determine which of those has more effectiveness and obviously safety. And then the extension programs will both include the drug that's proven to be more safe or has safety and greater effectiveness. Is that correct?
Yeah. That's correct, yes. We'll make the decision on which to proceed with for both the extension study and future clinical studies based on the data that we gather. And I think one of the things that we're trying to do with this program, and I think one of the things that Ed and his team were enthused by, is we're trying to make this data-driven, that we might have some prejudices going in about which we think is going to be better, but what we actually need to do is get the data, and then we can make a decision based on what actually turns out to be the best, and choose that.
Yes, thank you, Glenn. That's very helpful. So I'm sure you're both in discussions with both EMA and FDA. So which company is taking the lead on those discussions in the EMA or in the FDA, or are both of you taking the lead? Can you give us a little information around how that's working in the regulatory arena?
Yeah, I can start. So, as we said, Pat, we're working with the EMA to our plan, if agreed upon by the EMA, is to file by the end of this year. And so, obviously, our focus is going to make sure that we have our eteplirsen filed for marketing authorization in Europe. And what we've talked about in the agreement is that Summit will be responsible, for their regulatory submissions. And I think where we come in to help, we'll be ahead of them both in the U.S. and in Europe. We'll have valuable information that we will share. And I think, we're both small companies, and having more regulatory experience, because we also have people who have a lot of experience, not only in the United States but in Europe. And I think the way we're looking at this is we would combine resources to make sure that we have the best submission. But that for Summit would take the lead, and they would be responsible for the filings both in Europe and in the U.S.
Thanks, Ed. And Ed, in some of the interactions with both FDA and EMA, it's been mentioned that there can be parallel development or co-development on a particular drug, so that it might be approved both in the U.S. and in Europe at the same time. Would the Summit drug, and Glenn, this is probably for you, would the Summit drug, whatever compound in terms of F3 and F6, once that decision is made, would that be possible or of interest to co-develop it with EMA and FDA at the same time?
Yeah, that's a great question, Pat. And yes, our intention is that we would run trials that we would aim would give us the data for both a European approval and an FDA approval. And so we aim to have study designs that would give us data that would satisfy both regulators. And actually, in the Duchenne area, in the same way the companies are collaborating, there's pretty strong evidence that the agencies are collaborating as well. And so there's been quite a lot of congruence between discussions on endpoints and things like that. So, and I think it's a huge amount of encouragement from the patient and parent groups has facilitated that. So yes, I think we can have a a single development program that produces data suitable for both regional regulatory authorities, and that the trials will be carried out in both places in the same way like our phase out, the MD study is got sites in Europe today, and we will shortly be recruiting our first patients in the US. So it's a single study, but it's being carried out both in Europe and in the US.
Yeah, it's a single study in the U.S. And in Europe, and the timeline for opening in the U.S. is relatively soon, right, with a 48-week study?
Yes, it is. Actually, for those that have been following us, we're just a little bit behind. We got regulatory approval on track. It's just taken us a little bit longer to actually get all the sites, all the administration of the sites sorted out, and that's something in the future that we would hope Sarepta could maybe give us some guidance on, so we could become a bit more efficient on that. And I know we've had parents in the US who've been wanting to get into this study and been a little bit frustrated that we've not got going yet. But I hope that the tidal wave will be breaking over us very shortly.
Yes, it is frustrating for parents all around the U.S. and Europe, and in fact all over the world, to be waiting and knowing there's something there, a real possibility for their children in terms of clinical trial participation, and then having to wait on all of the IRBs and the contract negotiations and all of that before the sites get up and running. So it is frustrated, and we certainly appreciate you leaning on Sarepta's experience and trying to expedite that. We also have a question around the rights. And so the question is, why did Sarepta choose to pursue ex-U.S. rights versus U.S. rights, given where both companies are based? So especially since Sarepta's primarily regulatory expertise is in the U.S. So I guess this is for you, Ed. Is it better for Sarepta Sarepta to do U.S. and Summit to work with you on the conditional approval or the marketing authorization of the exon skipping technologies in Europe?
Well, I think the the agreement was for the really the commercialization rights in Europe, and we will be building out a commercial structure for Exondys 51, and we're in the process of doing that now. So we will have a commercial structure, and Summit really wanted to focus on the U.S. It's a little bit more of a straightforward commercialization because it's one country. So that was it's not so much for the let's say for the kind of strategic approach from a regulatory perspective. It really is more from a commercialization, and we will be able to commercialize this in Europe because we'll have a team in place based on what we're going to need to have for Exondys 51.
Yeah, thank you.
Yeah, I support that. And in fact, although we're both small companies and we happen to have our headquarters in the UK, and Ed's got his headquarters in the US, we are both really transatlantic operations. So already a lot of Sarepta's development work has been done in Europe as well as the US, and likewise our development work has been done in both places. So I think it's not really correct to think of them as a US company and us as a European company. We're really both in both places, and then it just comes down to the commercial realities of what's the appropriate and best partnership for both companies. And I'm extremely happy that Sarepta will have an effective marketing organization in Europe, and that it will really result in getting the product, if the data is good enough, into patient hands as quickly as possible.
Yes, thanks, Glenn. And I can't tell you how excited we all are for this collaboration and this development, because, as I say, these are two, in our view, foundational therapies to upregulate utrophin and to get a dystrophin protein expressed. So just the thought of that in Duchenne muscular dystrophy is incredible. So we're really excited about this partnership and really thankful. And I think in rare disease, we all know that this is an effort that has to be collaborative and global, and in order to reach all the patients. So if you wouldn't mind, I'd like to just talk about some of the current and planned clinical studies. And one of the questions that comes to mind as we think about combinations is, there are young guys who have completed the 12-person study, the original study of Sarepta, 201 and 202, and now are on, and many more are going to be on the approved exon disc 51 or eteplirsen. So one of the questions is, for parents and all of us, can they become candidates for this study in the U.S. in terms of izutromib?
Yeah, so I think in the long term, we are, like you, very excited about the possibility for combination treatments. But while we do know that exon disc 51 does work and has been approved, izutromib is still an experimental treatment, and so the first thing we need to do is show single-agent activity. And once we've proven single-agent activity, then we can move on to looking at combinations. So that means our immediate focus is on trials of izutromib on its own and comparing that to no treatment at all. So that means that patients who are currently on Exondys won't be eligible for this current study, and we'll concentrate on those patients who don't have an effective treatment at this point in time. But one of the big drivers of this deal is the potential for combination treatment once we have shown that izutromib is safe and effective, and we're quite excited to be doing that after we've shown the single agent activity.
Thanks, Glenn. It's just that, as you might imagine, for all families, we have long known that combination therapies are going to be incredibly important if we are going to halt progression of Duchenne. And so that's why the question comes up. And, of course, we'll be waiting so that these young men could be part of such a study or have access to the drug. So in line with that questioning, as you might imagine, when you're too young or non-ambulatory, you're often not included in studies. And what keeps me up at night a lot is the people who aren't able to join these studies, and certainly adults that are over 18 years old are never included in studies. So I wondered if and when you are considering studies in these populations. And Ed, I wouldn't mind if you answered that as well.
Sure. Well, I think you're absolutely correct, Pat, that when you do your first clinical study, it's always on a very homogeneous and narrow population because you try to control for all of the variables. But as we expand, so our focus is really to expand in other populations, and we have a number of commitments as part of our submission in Europe as part of the pediatric investigational plan. So we will be doing a study in younger boys and looking specifically at boys from 6 months to 4 years, which we currently haven't studied. We've only gone down to 4 years. And then there's a plan which will require some reformulation to even look at neonates. So we will need to understand that population, but we will be doing it through clinical trials. And so I think really the focus that we're trying to do is make sure we get drug as quickly as possible. And obviously the best way to do it, especially for the older population, is to get the drug approved. And in the U.S., of course, there isn't an age or disease severity limitation on the label, so it is possible to be able to treat these boys. And certainly by trying to get approval in Europe, that allows us to treat a number of older boys and really hopefully the whole spectrum of the population.
Glenn, do you want to answer that as well?
No, I think Ed's really just about covered it. I would say, though, that one of the consequences of the company, not just our companies, but all companies focus on this homogeneous ambulant group, has been that we focused in terms of endpoints on those things. And, I think you, the parent groups, have actually been quite effective, and I would encourage you to help generate data on suitable endpoints for the non-ambulant population and the older population, because when the time comes, that will help us terrifically if there is the natural history data, for instance, to allow us to do reasonable trials in these populations. But I think Ed's comment that one of the best things we can do for people outside the age group is get an approval, because then we can include people who aren't just in trials, but they can get the marketed drug, and that's got to be a significant advantage.
Yes, thanks, Ed, and thanks, Glenn. No, I know, and I totally agree with using a homogeneous population to understand in an outcome measure that you can rely on, whether that's North Star, whether that's 4-stair climb or 6-minute walk or whatever is identified. I think the worry is that these young adults over the age of 18 are really sort of out on the outside looking in, and that's why I asked the question, because we have the pool, certainly, and that was developed by Elizabeth Room's group and UPPMD and certainly Duchenne Parent Project in the Netherlands. So I think they've done a good job, and we are all working on patient-reported outcomes. I just worry and would like you to just keep in mind about this over-adult population and considering a safety study, because I think they've lived their whole lives waiting for this very day to come, and I want us to always be focused not only on them, but certainly on them as part of this community. So in line with that, there are policies in the U.S., certainly PDUFA 6 is being renegotiated this year. There are provisions in there to think about or to require companies to have a compassionate use policy. So basically, you may or may not know that J&J has led the way on this. The policy really basically states if and when, under what circumstances, and how that will be managed in terms of the request. We know that FDA has the ability, and in 99% of the cases, gives compassionate use when requested, and the company obviously has agreed to that. So I wonder if you are thinking about developing a compassionate use policy, and if you might be able to tell us your thinking along that line today.
Yeah, I guess the question's aimed at Summit, because yeah. So right now we don't have a compassionate use policy, but it is something that we've been considering very carefully, and really we need to get at least past the point where we've got some evidence of efficacy and a balance of safety before we do that. And yes, we are aware and are passionately interested in what's developing in the US and also in Europe as well in this respect, and not just compassionate use, but some of these early access schemes that are being debated and introduced as well. Because I think if any treatment is appropriate for an early access scheme, then the treatments for Duchenne would fall into that category. But right now, where we are with Azutramid, I think it's really important to keep patient treatment under the very watchful eye of the clinical trial regime until we're confident about the safety profile, until we've seen some signs of efficacy.
Ed?
Yeah, and I think right now, we also do not have compassionate access programs, and obviously our development is very complicated. And one of the reasons is we have a number of commitments for other studies, for dose-finding studies. We're still continuing with our boys who are 4 to 6 in our 203 study. As part of the pediatric investigational plan, we're continuing our 204 study, so we have to make sure that we have all of the regulatory commitments done, and really we'll be looking at that. And obviously, we're looking at things like even in certain regions of the world, we're still trying to figure out the freedom to operate, which does impact on what we're able to do. So for us, it's a fairly complicated managing supply and all of the ongoing clinical trials. But we continue to look at that, and I think our focus is, how do we get the drug to as many patients as possible and do it in a fair and equitable way. So we continue to look at that, and we continue to look at ways. And as Glenn said, the some of the early approval programs in Europe does give an opportunity that allows you to continue to manufacture. So we're looking at every potential possibility to try to get as much drug to patients as soon as possible.
Thank you both for that. I think it's so important. As you know, my sons are gone, but I can't imagine seeing this real hope and not having a way forward. So I appreciate you focusing in on a compassionate use policy or thinking about when and how that would look in the future. We recognize you have to establish safety, and we certainly wouldn't want to jeopardize any of your development plans at all. At the same time, we're really happy that you keep all of our sons, whatever age and whatever degree of function they have, in your head. So we're going to move on into current and future research. So as we think about combinations, and as you know, we would like all that to happen yesterday. So I wonder what is the FDA and the EMA going to require in order to combine the Summit Uzutramed and Sarepta's and your drugs that you're moving forward? Do you suppose that with every exon skipping technology they will require animal data or they will require some data? How will this work going forward? I'd love each of you to comment.
Well, I can start. I mean, obviously, I think Glenn correctly identified the issue. Is you have to know what the safety and efficacy of the drug is individually before you look at combination, and that's always a requirement. So I would say that once we understand what the safety and efficacy of etrasimod is, that then you could consider looking at it in combination. And the concern, of course, is there any greater safety risk? Is there any, increased efficacy, which would be the hope? Because we know based on the mechanism of action, we know that the dystrophin that we're producing doesn't take up all of really the sites that could be utilized by utrophin. And so from that perspective, it makes perfect sense from a scientific perspective to put the two together and to hope that they would be, there would be some synergy and that the combination would be more powerful. But all those things have to be done, and we have to kind of work it out with the regulatory authorities. I would hope, the nice thing about looking at utrophin upregulation is that it's really the same mechanism, and it's going to be the same drug. So it's not like it's a personalized genetic medicine. It can be used potentially for any patient with muscular dystrophy. And so I think once we have more information on our follow-on exons, I would envision that it wouldn't be a very complicated pathway. Once we have multiple exons that are approved, I would assume that, being able to combine it, once we have the safety and efficacy of utrophin upregulation, then I think that would make perfect sense. Glenn, any comments?
Yeah, I agree with you, Ed, and I wouldn't be too concerned about what the EMA and the FDA say, because in many other areas, cancer, for instance, there are pretty well established routes for getting combinations approved. So I think the most important thing is for us to show single agent activity of etrasimod, and then I think the route for looking at combinations is fairly straightforward, but it does have to be in that sequence of showing the single agent activity first, and then I think the trials will be relatively straightforward, and the regulator, again, will be relatively straightforward.
Right, thank you. And just in the really enthusiasm of the community to try to move this forward, I dream of the day when both the exon skipping technologies for multiple exons are approved, as well as etrasimod are approved. And I can imagine going to your clinic and asking for both of these drugs. And so one of the questions would be, we're going to need to know drug-drug interactions. We're going to need to know if one dose impacts the next. So I just wondered if there are any plans to do some animal studies around that on one of the mouse models or a dog model, or for that matter, one of the pig models that they're bringing forward. Have you thought about? You probably have enough on your plate right now today without thinking about this, but just jumping ahead, are your scientific teams kind of looking at what do we need to know, what do we need to do, what do we need to learn about this in the future as we go forward?
Yeah, they certainly are. In fact, and this was one of the collaborations even before we entered into this agreement that our team were excited by, and then we got a call from Sarepta's team saying, why don't we have a look at this? So I know both teams are excited to devise the appropriate animal experiments to look at this. I mean, the work has not actually started yet, but it is something that we're likely to see well before we see any clinical data.
Yes, and that was, I think that was what really interested it. It just seemed to be a natural marriage using the two products. So we will be doing those animal studies long before we do it in human.
Yeah, no, I think that will be exciting, and we as a community will look forward to hearing what you're learning in the various models that we can sort of look forward to the day when both of these technologies are approved, and we can see what to expect and perhaps what to see and pin our hopes on in the future. So back to one of the more difficult subjects, and that is biopsies. Biopsies are included in the Utrophin study, and so as you know, this is really difficult on children, on their families. It's just difficult for us as a community, and we were faced with some of the challenges that you had experienced throughout the analysis of dystrophin in the 201-202 study. So we wondered, are you using the same or similar methods for quantification? Are these validated in terms of understanding utrophin and its expression and its distribution across the membrane?
No, these are really key questions, actually, Pat. And again, this was one of the collaborations that we've had with Sarepta for some time on stretching from how you actually take the biopsies in the first place, and then the technologies for examining what's going on in the muscle as well. We've got an additional layer of complication over and above the issues that exon skipping has with dystrophin, and that is that a boy with DMD has no dystrophin almost by definition, and so if you see some dystrophin, then that's a great win, a big step forward, notwithstanding the debate with the FDA about just how much dystrophin you need. But with utrophin is produced naturally by everybody in response to injury. So not only is utrophin produced in early muscle development when the muscle's first laid down, but it's also produced in response to injury. And of course, boys with Duchenne, their muscles are constantly going through a cycle of repair, then maturation, and then failure again. And during that cycle, they'll make utrophin during the repair, then the utrophin will be turned off, no dystrophin arrives, and so the fiber is vulnerable to damage, then it is damaged, and then utrophin is turned on. So there are relatively high levels of utrophin in the biopsy samples from DMD boys, but it's very patchy. So what you see in a biopsy sample is one area of fiber that has a lot of utrophin, because that indicates that there's been a recent failure and there is an active repair going on. And then you'll see other areas where there's a little utrophin, and that's because the repair is maturing. It's been some months since the repair, and then there'll be areas that have no utrophin at all, and those are areas where the repair was quite some time ago, and these are vulnerable to catastrophic failure again. And if our treatment is working, what we would expect to see is a more uniform utrophin, so that the izutramid is encouraging the cells to produce more utrophin even after the repair is complete. So we're really looking not just for more utrophin, we're looking for how that distribution of utrophin is more uniform. And so success for us is going to be slightly more complicated. And, we've had a lot of encouragement from Sarepta and their team, and we've been working with various outside vendors for quite some time, and actually just published some data at the World Muscle Society on how the validation of this utrophin methodology is going on. So that was a long answer, but your question is, it's more complicated to measure utrophin, but we've been working with a number of academic institutions. We've actually had some great financial support from Joining Jack, one of the UK patient groups, towards this, and we were steered towards what has turned out to be a great company that specializes in these called Flagship Biosciences, and that was an introduction by Sarepta for us, which has worked out very well to develop automated techniques to try and validate this uniformity of utrophin. I think one of the things that we hear from parents who've been involved in the beginning of our phase-out DMD study is that the amount of tissue taken in the biopsies is quite large, and I think what we've been trying to do is work to minimize the invasiveness of it. But right now, with the state of technology, we do need to ensure that we've got enough material, enough muscle to really see the effect of these drugs. And what I would hope over time is that these techniques can be refined so the amount of tissue required is less. But given the state of development right now, we really do need these biopsies. It's an essential part of finding out whether the drugs work or not. Rest assured, we're working as hard as we can to minimize the impact on the boys of these biopsy samples.
Yes, thanks, Glenn. It seems to be very complicated, certainly when utrophin is called in to help with the repair, and then lessons when that repair is taking place and utrophin doesn't fill in the blanks. So could you tell us a little bit more about then, you're taking a bigger sample in terms of the biopsy, and obviously it is an invasive procedure. Can you give us a little more insight? Does it matter? Are you imaging so that you see a portion of the muscle that you wanted to take so that it's less likely to have a great deal of fat and fiber in it, and more likely to have muscle so that you can really examine the tissue well? Can you give us shed a little more light on the biopsy procedure itself? And also what we've learned as a community, sometimes in various clinical sites, surgeons are different, and there is perhaps less knowledge about the biopsy sampling itself and what kind of quality of tissue they need. So could you give us a little more information around what you're doing to make sure that these biopsies are taken, that you have the kinds of cells necessary to understand what if utrophin is upregulated, and not just in times of repair, and that you can assure yourself that the cells you're taking are going to be useful in terms of interrogating?
So maybe I can start out, Glenn, because we've had to deal with these issues for quite some time. So I think one of the things, Pat, is if you have the right entry criteria. And what we've seen is that, if you have a fairly homogeneous population, if you're using upper extremity biopsies, the amount of atrophy and connective tissue is certainly reduced than what you would get if you were looking at such as a lower extremity muscle like the quadriceps. The other thing that we've been doing, and obviously something that we're going to be sharing with Summit, is that we do an H&E stain of the muscle biopsies to make sure that it's predominantly muscle and not other tissues. And in regards to training the sites, you're quite right that the sites can vary. But what we've put in place is we have very specific surgeons who are trained on the technique, and each site can have slight differences in the way they do the biopsy. What we do is make sure they do it in exactly the same way, using the same procedure, if it's a muscle clamp, and doing it exactly the same way every time. And we have site visits to the surgical sites. We even have them send, it's actually a piece of steak that they have to process and obtain, and to show that they know how to cut it and they have the right amount. And we do that a couple of times before they ever do a human sample. So I think what we found is that if you have a very rigid protocol, it's a little bit like a detailed recipe that you'd use for cooking. If you follow the recipe, it will be good, and you just need to make sure that there's a few dry runs. And so it's a lot of effort, and it means having to audit the sites and having to make sure that everyone is trained, having protocols. But if you do it that way, it does reduce a lot of the error and the problems that had been seen in other studies where you do biopsies and the results are uninterpretable. So far, from what we've seen, and I would expect that the summit will follow procedures that are very, very similar. You can reduce a lot of the error and the problems, and hopefully we'll have better tests. I know Summit is doing the MRI, and we're doing it also. Right now, though, we have no idea what utrophin upregulation or dystrophin production does on what an MRI is, and that's a very distant surrogate marker. So we really do need to understand what's happening with the muscle, and then hopefully we may be able to extrapolate and use smaller and smaller samples or even non-invasive techniques, but we're just not there right now.
Right. Well, honestly, Ed, we were really pleased the way you did the studies in terms of limiting the number of making sure the surgeons were fairly consistent in the process and procedure consistent, and we appreciated the analysis at 2 specific labs because I think that does limit, as you know, as we sort of are learning to fly the plane as we're in the very early days. It was disheartening to learn some of the biopsies that were taken weren't useful, and I think. I won't say the community is happy to give biopsies. There's no parent in the world that wants their son, and probably no son who wants to give muscle. But we certainly understand the need to evaluate muscle. We just want to make sure that it's done thoughtfully and carefully and that you get the best answers possible so that we can expedite the approvals with you and take advantage of these really promising opportunities that are coming our way these days. So thank you for that. And know that we as a community will always work with you. We're thankful for this partnership. We think it's amazing and wonderful and really brings hope that we've wanted and waited for so long. And I know both of you, in particular, you've been running around for months and months and months with the approval of eteplirsen and then certainly the partnerships and then World Muscle Society. And probably neither of you had any sleep for months. But we wanted you to know, both of you, Glenn and Ed, we so appreciate your efforts. We hope that you'll continue to convene this community. We're thankful for the information you give us and for your transparency and answering questions, even the hard ones that we ask, and know that we recognize who you are and what you're doing for us, and we're thankful you're a part of our community. So thank you very much for this call today. We look forward to more communication, and hopefully frequently in the future we will touch base and learn more. And also know that this community offers our full support to you, no matter what that is and what that looks like. We're behind you and thankful. So thank you, Glenn. Thank you, Ed, for joining the call. It's been a pleasure.
Thank you, Beth. This is as always. This is also informative for us to talk to you.
Yeah, we think so too, and also to parents who are maybe listening on the call. Continue to email us or write us or call us. So we can't always answer all your questions, but we'll do our best.
Thank you. And this call is recorded, so it will be posted online. Thank you both again.